Good morning. Hello. Hello, hello. Okay. Hello.
Good morning. If you can quietly sit down and we can get started. Well, welcome to the second day. I've the honor to introduce our early career keynote speaker this morning. And so, Okay, so yeah, I'm very excited to introduce our early career keynote, Dr. Cusa today. Marine is a marine policy advisor with the Oceania's anti-illegal, unreported and unregulated fishing team, dedicated to combating illegal fishing and improving seafood traceability in the EU. Marine actually grew up on a sailboat, which inspired her lifelong passion for the sea. With a PhD from the University of Salford, Marine's expertise lie in using molecular tools for seafood traceability, ensuring the authenticity of catch locations. Her work bridges science and policy, collaborating with academics, NGOs, and the FAO. For those of you who attended the workshop on Sunday, you will also know that Marine is a great science communicator, and she's running a popular YouTube channel and regularly contributing to podcasts and articles. Today, Marine will be talking about DNA-based methods and their role in enhancing seafood traceability, addressing challenges of mislabelling fraud and the need for innovative solutions. Please join me in welcoming Marine Cusa.
Thank you, and thank you so much, Elena, for this very kind introduction. Thank you all for being here. It's really an absolute privilege. I'm really, really happy to be given this opportunity, and thanks to ICES as well for inviting me today as a keynote speaker. So I wanted to start this presentation with a little story because I think it does help put things into perspective, and it's also entertaining. When I was an undergraduate student, which was more than a decade ago, one summer I had an internship in France where I worked on bottlenose dolphins for a few months, which I thought was pretty cool. And as part of that internship, I had to board a commercial fishing vessel to sample fish for stable isotope analysis. And I just immediately forgot about dolphins. I completely fell in love with everything that had to do with fisheries. The sheer complexity, the ecological complexity, the economical, political, everything that had to do with fisheries was just fascinating. And so I became very good friends with those fishermen, and as they started trusting me, they also started doing things that were somewhat questionable. I will tell you one of these stories, but I do want you to keep in mind that they are very kind people, and I am still very good friends with them today. And but unfortunately, sometimes they do engage in activities that are, as I said, somewhat questionable, and it is a problem that we need to find a solution to. So one evening, we were at sea, and we weren't catching anything, which of course was my fault because I'm a woman, and they made a very good point of reminding me that a woman is bad luck on board. And however, at some point we caught this huge haul of rays. And the fishers looked a little bit flustered, but they started processing the rays nonetheless. They cut the wings, threw the bodies back in the water, which is often what they do with rays and skates. And the captain came to see me and said, "Look, Marine, you have to keep quiet about this. We're not allowed to land and sell these rays." That was before the landing obligations. And I don't remember the species, unfortunately, because it was more than a decade ago. And I thought, okay, well, yeah, you do your thing, and I'm just going to keep my mouth shut. In the morning, when we went back to port, the captain got a call from one of his colleagues who said, "Look, the authorities are in the port, and they are checking the halls. So if you have anything fishy down there, watch out. Of course, our hall was full of fishy stuff and illegally caught rays. And so instead of going to port, we went to a dock that was just outside the port. There was a truck waiting for us there. They unloaded everything at record speed, and then we went to port. And of course, we had nothing to declare because Marine was on board. Bad luck. So I think this is a good segue into my talk, where I'm going to talk a little bit about how advances in molecular tools can improve seafood traceability. But before I do that, of course, I want to talk a bit about seafood traceability itself, define it, what is the context right now, and how molecular tools have been used already in the context of seafood traceability. So I'm sure a number of you have seen this figure. It's being published on regular occasions by the FAO. This is the latest one. What I did want to mention is that before understanding the context of seafood traceability, it's important to understand the seafood consumption landscape. And this figure represents the seafood production over the course of a few decades from the 1950s. And you can see that it's been steadily increasing thanks in large part to aquaculture. Now, this increase is obviously directly related to an increasing demand, right? We wouldn't need to produce so much fish if there wasn't as much demand as there is. And in fact, there are more people on the planet, but our demand for seafood is also increasing per capita. So we are more hungry for fish than we used to be. Now, I just want to draw your attention on the blue lines, which represent wild capture fisheries. And you can see that ever since the 80s, it's been quite constant. It's a bit plateauing, let's say. And so it can give the impression that we've been harvesting sustainably and that every year we harvest the same amount of fish, which is good. But the reality is that this also hides a problem. Not all fish stocks are being harvested sustainably. Some of them are, but many of them are not. And as a result, because of stock collapse and unsustainably harvested stocks, we have to fish an increasing diversity of fish. So we have to fish deeper, we have to fish further, we have to fish smaller. And this increasing diversity of fish also contributes to an increasingly complex seafood supply chain. In order to deal with such a complexity, and there are many reasons why the seafood supply chain is complex. The diversity of species is one. The fact that seafood is one of the most traded, in fact, it is the most traded animal protein in the world. And it also has a very complex regulatory framework. This all makes for a very difficult traceability landscape. And it is, of course, traceability is definitely one of the key elements to manage this industry. So before I move on, I just want to make sure that we're all on the same page here with the definitions of transparency and traceability. They're often used interchangeably, but they're not exactly the same thing. Transparency is the availability of accurate information, and traceability is the transfer of that information from one level of the supply chain to the next, ideally all the way up to the consumer. I am quite familiar with the traceability and transparency landscape in the EU, unfortunately. I know a lot of you come from outside the EU, and I don't know what the regulations are outside the EU, but what I can tell you is that in the EU, it's not too bad. There's some pretty strong regulations, and you can see this this chart here was published by Sea Choice, which is a Canadian NGO, where they compared the labeling scheme. So obviously labeling is directly related to traceability. They compared it between the EU, the US, and Canada, Canada being the worst. I am part Canadian, so I'm a little bit embarrassed. And the EU being quite good, as you can see there. Now, this is true for fresh products, but there are many, many products, seafood products in the EU that still fall through the nets, like canned, processed products, for example. And in fact, yesterday I went to a supermarket to look at the labels of a few cans, and it doesn't look very good. I invite you to do that. It's one of my favorite activities to do on my free time. So, in order to achieve adequate traceability, there's a certain number of elements that need to be aligned, and unfortunately, they don't align super well. So one of the first problems we have is that a lot of the traceability of seafood is still largely paper-based in the EU and also elsewhere in the world. Now, with the revision of the fisheries control regulation in the EU, that may change. But at the moment, there's still no mandate saying that traceability should be electronic. So some of it is electronic, but it's really up to the business owners themselves to decide what traceability system they're going to use. And that leads me to another point, which is that in the EU, each member state is responsible for implementing the regulation, which means that there's a lot of inconsistencies. Some do it well, some don't do it super well. And business owners themselves are responsible for acquiring the adequate traceability methods, again, leading to quite a lot of inconsistencies in the industry. So with this, I would just like to move on to talking about the way molecular tools have been used in the context of seafood traceability. Now, it's often said that DNA-based tools are a traceability tool. They're not exactly a traceability tool because they don't enable traceability to happen. They're a transparency tool, which means that they're often used to verify a claim at a point in the supply chain. And they've been used a lot to uncover issues of mislabelling. They've been used a little bit for enforcement and deterrence. And I will go in each of these points more in details. When it comes to mislabelling, I'm sure you've heard about it. It's made a lot of noise over the last decade. It started, the use of molecular tools to uncover mislabelling started roughly in around 2008, with some first studies being published in the US and in Canada, and uncovering huge amounts of seafood mislabelling. And then quickly that spread to the EU, and eventually it just spread like wildfire around the world, showing that mislabelling is really a problem everywhere, globally. And of course, it got picked up by the media because it's an issue that affects consumers directly, and consumers don't like that, and the media like that. So we had hundreds of media articles on this issue. Before I move on, I just wanted to go through a little exercise here. So these are commonly consumed species of fish in Europe. And if you know what they are, then that's great. You can try and guess what they are. I'm not going to stop here to give you the time to do it, so you have to do it while I talk. And the reason why I'm showing you this is that we did a little exercise where we looked at the knowledge that consumers had of the seafood they consume. Can the consumers actually identify the seafood they consume? And hopefully you're doing better, at least if you're from the EU. If you're from outside the EU, I'm not blaming you. Otherwise, I don't know, it's a different story. The consumers are doing really, really poorly. Their ability to identify really common species of fish is pretty bad. We had a 30% correct identification, and this study was conducted across 6 countries in the EU. The UK was part of the EU at the time, so bear with me here. And we interviewed about 360 people. We accepted all sorts of answers as correct. Sole, salmon, all of this was accepted as correct. And of course, we also used local languages. So this goes to show that there is a bit of a problem. Consumers are a little bit detached from the seafood they consume. They consume seafood without necessarily knowing what it is. And I think it was the problem is summarized very well by Ian Urbina in his beautiful book, The Outlaw Ocean, where he says, an entire ecosystem that encompasses millions of species of creatures is lumped together in popular consciousness, consisting not of distinct animals, but of things we consume. Now, the reason why I'm mentioning this is that it is difficult to hold the industry accountable if the consumers don't seem to care themselves. why should the industry care if the consumers don't care? I decided to do a little exercise here, partially because I was very curious. And the other reason why I did this is because it's extremely trendy. And whether you like it or not, you have to agree it's trendy. I asked AI what it thought of the concept of seafood mislabeling. And not only that, but I asked Midjourney, which is an AI image generator. So I said, imagine the concept of seafood mislabeling. Now, I don't know about you, but I have no idea how I would illustrate that. It's very broad, and it's such a complex concept. It's a bit of a daunting task. But within a few seconds, I came up with this. Now, this is a chef, clearly, who is leaning over a pile of crabs. They look a bit strange because AI has some problems with details. And he's swimming in money and coasters for some reason. But again, AI is imperfect. But lots of money. And so what this image here is illustrating is clearly someone who is committing fraud in one way or another and benefiting from it financially. And indeed, AI here seems to be associating mislabeling with fraud. Now, it's important to understand that mislabeling and fraud are not, they don't mean the same thing. Fraud can lead to mislabeling for sure, but not all mislabeling is fraud. So I have to tell AI that he needs to think a bit more about this concept of seafood mislabeling. There's partially because of the complexity of the trade I was just talking about, there's a lot of errors that happen. There's also other issues like social issues, poverty, that can lead to problems of mislabeling. So now I just went a bit on a tangent. I'm going to go back to molecular tools. Molecular tools can also be used as deterring tools, especially if they are used on a regular basis. Now, of course, if they're used once a year, it's not going to deter anyone. But if authorities are using them regularly, once a week, or maybe even once a month, it can function maybe as a deterring tool. And these people are enforcement officers in Indonesia who work more specifically on identifying shark products. And Indonesia being one of the largest exporters of shark products, it is very, very difficult to identify shark products when they look like this. And often, by the time they are being exported, they look like something like this. So they're exporting a lot of fins, but they also export skin, they export shark powder, and these are trunks that are devoid of any morphological features. And even to highly trained shark taxonomists, it can be really, really hard to identify. So they had a huge interest in using DNA-based tools to, on a regular basis, to deter malpractice and to identify species that are being exported. This is in a storage center where the products are being stored ready for export. And of course, the other context in which it can be used, molecular tools can be used, is in the context of enforcement. Now, these things are they kind of work together, right? Deterrence, enforcement leads to deterrence, etc. It's not clear. There's not a clear-cut boundary between them. And I really wanted to talk about this example, which I think exemplifies that quite well. And it's an example that was published by Diego Cardeñosa in 2019. He had developed a qPCR assay to identify European eels. And for those of you who don't know, European eels are endangered, and there's a ban on export and import, in this case in Hong Kong. And the Hong Kong airport authorities called Diego Cardeñosa, who at the time was a PhD. I wish people called me when I was a PhD. And they asked him if he could come to the airport with his device, his qPCR assay and his qPCR device, because they had caught some people who were bringing in some glass eels. So glass eels is a juvenile stage of eels. And there are many species of eels, most of which are fine, and they can be exported and imported. The problem is when they look like this on the right, when they're at that stage, it's very, very difficult to tell them apart. So Diego Cardeñosa went to the airport, and within a few hours, he was able to identify that they were all European glass eels, endangered. And so the smugglers were prosecuted. So this is a very good example of how it can be used in the context of enforcement. Now, despite this, there is quite a divide between what researchers provide, which is evidence, and what authorities demand, which is tools to solve the problem. There is, and to give researchers some credits, there is quite a lot of research out there that is done to try and design tools like qPCR assays or real-time PCR assays that tends to be usually species-specific. But often, these tools are shared amongst ourselves. They're presented at conferences, they're published in peer-reviewed papers, and trust me when I say that authorities do not read these papers, they don't have the time, and they don't go to these conferences. So how are they supposed to know that these tools exist? There are some projects, like the Label Fish Project, or the Fish Pop Trace Project, or the Sea Traces Project, which is the project as part of which I conducted this research, that try to bridge this gap between academics and stakeholders and authorities. But again, there's still quite a lot of confusion as to what exists out there, what can authorities use themselves. So what are they demanding? The moon, that's what they're demanding. They're demanding tools that can, first of all, ideally, a lot of them have expressed interest in universal species identification tool. So a lot of the tools that exist that are easy to use, like qPCR devices or tools, are species specific, meaning that you can use them with one species or a group of species, but that's all. They want something that you can use with every species. Then they want something that is easy to use. They can use it themselves. They don't need any expertise. That is quick and portable. So basically, they want a phone that you use, you scan it, and bam, you get an answer. Obviously, that's probably not going to happen, but we can try and come pretty close to that. And then, if possible, they would also like things that can go beyond species identification. So conveniently, when I started my PhD, our lab got approached by a private company called Ecologenics, who had designed this qPCR assay. And they claimed that it was portable, rapid, cheap, easy to use, and, lo and behold, universal. So basically, it was everything we were looking for. It was perfect. And it seemed a little bit too good to be true, but, you know, who knows? And I'm not going to go into the details of how it functions at the molecular level, but if some of you are interested, I am more than happy to talk about it afterwards. It is a qPCR assay, but unlike most of those you've heard about who are species-specific, that one uses a set of probes that creates fluorescent signature for species, and it works for all the species out there. It's somewhat universal. At least that's what it claimed. So we embarked on this journey to test the method. The company sent us with another set of, two other labs, one in Canada and one in the UK. We tested 75 samples that was sent to us by the company. So it was a blind study. We did not know what they were. And they belonged to 18 commonly consumed species, fish species. And we had a 98% success rate in identifying these samples, which is quite high. And most importantly, we had no false positives. And that's really important, because if you're going to use a tool like this in the context of enforcement, you don't want a false positive. You don't want to falsely identify something as something it's not. So that was extremely promising. And we thought, wow, this is a step forward. But the samples had been tested by the company before, and they had been sent to us by the company. So we decided to take it a step further and test this method on board a research vessel, the CFAS Endeavour, during a pelagic survey that they were conducting in the Irish and Celtic Sea and the English Channel. We brought the method with us. So on the right, you have my little bench lab. They gave me a space in a fridge, the beauty of being a PhD student. And every time I had to do my analysis, I would set up the lab, and then I would clean up the space afterwards. So it was really easy to set up. And now the method has been optimized so that it doesn't even need pipettes or vortex or centrifuge anymore. So all you need is the MIC, which is the PCR machine here, and a computer. And we sampled fish opportunistically. So it was a pelagic survey. We had a lot of pelagic species in there. And all the species we tested, none of them were in the database of Ecologenics. They had never been tested by the company. And it was really interesting because out of the 15 species we tested, we managed to generate 14 species-specific signatures. So I'm just showing you 3 of them because it does get a little bit messy when you look at 14 of them in there. But this is an example of what it looks like and how visually compelling it is. And also how different they are between species. What the method, the company uses an algorithm with an online database they create in order to get a success rate and also to be able to identify the species. So instead of identifying them visually, which can get pretty messy, you actually have a software that tells you what it is. And of course, because none of these species were in that software, we had to add a few. So we worked with the company, we added 5 of them in there. It's quite a long process. And we had about a 70% success rate, which is still pretty good. And again, very important, we had no false positives. But as you can see, the success rate was a little bit lower than when we tested them in the lab. So why is that? Well, interestingly, the method itself targets a very small chunk of the CO1 barcode. And it's a mini barcode, basically, of about 80 nucleotides. In order for the method to work, this barcode needs to be exactly the same for all members of a species, and it needs to be different between species, because each of these barcodes that's different will create a different fluorescent signature. Now, there are some species, like whiting, that have 1 nucleotide difference. They have several variants within that mini barcode. So if that's the case, the method is so sensitive that it will actually pick that up, and it's going to give you 3 different fluorescent signatures for a single species, and that's obviously a problem. So you need to account for these variants in your database when you are doing your analysis. But the method itself, in the end, was quite promising. It seems to be universal to some extent. We even tested it in Indonesia with extremely processed, heavily processed shark products, fins that had been dried, that go through chemical processing, and it still works. It's very impressive. It's not as, it doesn't work as well for cartilaginous fish, so it probably needs to be optimized for sharks and rays. But when it comes to bony fishes, it doesn't seem to misassign any species. So that's a very good point for the method. And of course, it's rapid and it's really easy to use. On the other hand, obviously, it's designed by a private company, like many methods. But yeah, so you are dependent on that company for if you want to use this technology. Because we wanted to test a number of methods here, we also decided to work a little bit with Oxford Nanopore Technologies. Now, this is completely different. The method I just talked about is a qPCR method, and it's kind of the same as when you're doing a COVID-19 test. It's very simple, it's very quick, but it doesn't sequence. On the other hand, Oxford Nanopore has developed these tools, so this is a MinION, that are sequence. They can sequence, in fact, they can sequence the entire genome if you want. So the kind of questions you can ask and answer with a device like this are much, much more complex. In fact, it seems a little bit sad to just use that for species identification. You can really ask things that are much more complicated. You can do mitogenome assembly. You can look at geographical provenance. I mean, the use really is infinite. But because it is a sequencing device, it is also more computationally intensive. And you do need a little bit of bioinformatics knowledge. So it's hard to sell to authorities unless they have someone who is specialized in using molecular tools. So we, along with the project C-Traces and the Mariani Lab, we demonstrated these tools to authorities, in this case the Marine Management Organization in the UK, and the Sea Fisheries Protection Authorities in Ireland, who are very interested in using that in the context of enforcement. And in fact, the MMO has already purchased some MICs at the magnetic induction cycler, which is the little PCR device. It's that red box. And they're very interested in using that in the field. So this is all great, but one of the questions I got more than once was, you can tell me what this species is, but I can tell you what this species is. I don't have a problem identifying it. I'm a taxonomist, and this is easy. What I want you to tell me is, where does it come from? Now, this is much more complicated. You can use molecular tools for that, of course, but you need certain conditions to be aligned. It's not as simple and straightforward as identifying species. First, what you need is biologically segregated populations that match your management areas. And that is not always the case. We know that. Sometimes management areas overlap with several populations or one population, or vice versa. So you need to meet that condition in order to be able to use tools to assign individuals back to a particular area, in this case a management area. What you need as well is enough genetic differentiation between your populations in order to be able to tell them apart. So you need some form of reproductive isolation. If there's too much mixing between the populations, again, you might be able to assign an individual back, but with pretty low certainty, and that's a problem in the context of enforcement. And ideally, you'd want your species to be relatively sedentary. You can use this on migratory species, but again, it's a bit more difficult. What we went on to do is to look at the use of molecular tools in combination with stable isotopes and see if using these tools together improves your chances of assigning individuals back to their population of origin. And we worked with specialists in stable isotopes to do this. And what we noted is that combining methods doesn't necessarily improve your chance of assigning an individual back to a specific area, but it severely decreases your chances of misassignment. And again, that is very important. Basically, it decreases your chances of having a false positive. When you are using a tool like this in the context of enforcement, that's what you want. You don't want false positives. So it can be quite useful to combine methods like this. What we went on to do is to actually test molecular tools for geographical catch location in the context of mislabelling, which is not usually done. Often, in fact, pretty much all the time, mislabelling is used at the species level, but it's rarely used beyond that. We chose cod because cod is a very well-studied species with distinct populations. There's a lot of genetic studies on cod. And more than that, there's also assignment studies. These are studies that have been able to assign individual cod back to their population of origin with extremely high certainty. So then we went on to sample cod around Europe, again, I want to say the EU at the time. The UK was part of the EU. In France, Spain, and Germany. And as you can see, looking at labels is really one of my hobby. And we looked at the geographical catch location that is indicated on the label. And then we tried to see if we could assign cod back to their population of origin. So in this case, we were looking at the Northeast Arctic versus the North Sea. Of course, you have other cod populations in Iceland and Faroe Islands, but we weren't focusing on those. And we came up with these results. So I'm just going to walk you through this slowly because it is a little bit confusing perhaps. So I'll ask you to focus on just on the figure here. This is a canonical, this is a correspondence analysis, sorry. And it basically smooshes your results in a two-dimensional space here. But of course it's a multivariate analysis, so we're only seeing part of the results here. In yellow, on the left, are all of the Northeast Arctic reference samples. So we know they come from the Northeast Arctic. And in blue, on the right, are all the North Sea reference samples. And we know they come from the North Sea. And you can see they do cluster quite nicely. Now, there is some overlap, which is most likely due to the fact that we are smooshing this into a two-dimensional space. But nonetheless, I think it still looks quite compelling. Then what we did is that we overlaid our market samples on this. The letters correspond to what was written on the package. So you can see that almost all of the samples that were labeled as coming from the Northeast Arctic cluster on the left, which is good. That's where you want them to be. That is where the Northeast Arctic reference samples are. But there are a few here, in red, in fact, that are labeled North Sea. And in fact, using some Bayesian models and probability tests, we were able to show that these are the samples that are mislabeled. So those samples on the left that are in red happen to have been assigned back to the Northeast Arctic. So in fact, they are Northeast Arctic cod that were mislabeled as coming from the North Sea. Now, someone asked me, why would you want to mislabel a cod coming from a sustainable population? Why would you want to mislabel it as coming from an unsustainable stock? Because the Northeast Arctic is not, the North Sea cod is not doing so well. Well, I can tell you exactly why. When we were sampling in the UK, and we would ask fishmongers, where does this fish come from? They would say, oh, it's local. It was fished right here in the North Sea. It's just because they want to show that it's local. So that is why you have this kind of mislabeling, probably. There might be other reasons, but I can tell you that is one of the reasons. And because consumers want to eat local. So with this, we come to the end of the presentation. And I just wanted to go over some of the take-home messages again. Molecular tools have been unable to cover quite a lot of instances of mislabeling, which reveal that there is an issue somewhere in the supply chain and with the traceability system, which really also kind of illustrate the complexity of this industry. And enforcement and systematic verification are crucial compliance and deterrent tools. But unfortunately, there is a bit of a divide between what researchers provide, which is evidence, and what what authorities need, which are enforcement tools. Now, as I mentioned, researchers do occasionally provide tools that could be used by authorities, but it's not really communicated with those authorities. So there's a communication problem here. And what I've heard, now that I work with an NGO, I've heard quite a lot of stakeholders. I work with a lot of members of the industry, the seafood industry, and policymakers. And I've heard a lot of people say, oh, molecular tools are great at showing that there is a problem, but they're not reliable in the context of enforcement. And that is just not true. So I really hope that with this presentation I was able to show you a little bit how they can be used in a practical context. And I thank you very much for your attention. And also wanted to say, Eskerik Asko. Thank you.
Thank you so much. That was an incredibly engaging and interesting talk. Are there any questions? Yeah.
Well, first, thank you for it was indeed a very good talk and a very engaging talk. What I wanted to ask was concerning the last bit of info you presented about the origin of the fish. I was wondering because sometimes maybe the fishermen are catching on an area like the Northeast Arctic and there's migratory fish coming from the North Sea, for example, or vice versa. And in that case, they will still be labeling the fish as coming on the area they fished it, even though the fish is not from there. Can you actually say that this is mislabeling?
Yes. Well, obviously, this is one of the questions we asked ourselves when we worked on this, and this is why I was saying, if you're working with highly migratory species, it's a bit more difficult. It's not impossible, but it's more difficult. There's several answers to that question. First, when it comes to cod, these cod populations don't tend to mix. The North Sea and the Northeast Arctic, particularly the Barents Sea cod population, they do migrate down south to the coast of Norway for reproduction, but they don't usually go as far south as the North Sea, and the North Sea cod doesn't usually go back up. It could happen, but if it did happen, it would probably be on really rare occasions, and in this case, I don't suspect we would see that much mislabeling. And in fact, I didn't mention it, but we observed the 30% mislabeling rate of the catch location. That being said, I do think that in order to avoid these kind of problems, if you are going to use such a tool in the context of enforcement, first, you would want more than one sample. And the problem when you're doing an endpoint analysis like this, which is, by endpoint, I mean the end of the supply chain. At this level, we're really taking fish from fishmongers and supermarkets. You can only have one sample because you don't know if the fish in the same package next to it comes from the same haul or not. So this is a problem when it comes to statistical certainty. If you are, on the other hand, using that method at a landing site, and you can sample many, many, many individuals, then you will, of course, have a much higher certainty. The other thing is, if you are using this in the context of enforcement and you're actually going to prosecute people, you can't use just molecular tools. You have to combine that with other tools. You have to combine that with logbooks. You have to combine that with perhaps stable isotope analysis. So this is to show that it is possible. But again, there is some uncertainty, and you have to be really conservative when you interpret the results.
Thank you.
I see some people fighting, so we might go for the... Yes.
Hi,
and also, sorry, when you ask a question, could you please introduce yourself? Yes.
Thank you for that reminder. Thank you for a very good presentation. I'm Peter Edwards. I'm from the Pew Charitable Trust, another NGO. My question relates to, I guess it relates to, and I'm very interested that you used the AI tool. How do you think machine learning, AI, and all of those tools can help kind of consolidate some of those mistakes, including that slide that you had, you could have the 3 phosphorescent signatures for one species. That's the part one. How do you think AI generally, and machine learning, can help improve that and hence fine-tune it? And also related to that, the democratization of the information. You know, a lot of this stuff you said is owned by a private company. how do you see machine learning and perhaps open source types approaches of making sure that this information is broadly available to a wider community?
Yeah, thank you for this question. So I'll be honest with you. Machine learning is not my field. However, interestingly, when we tested this this fast fish ID method in Indonesia, the company Ecologenics on sharks, we tested it. The company they had never tested it on sharks, and they don't have any sharks in their database. So the person who was leading this project, Andika Prasetyo, who is from Jakarta, he actually used machine learning to create his own database of sharks and to assign the sharks back, and to identify the sharks using an algorithm of his making. So machine learning is definitely extremely useful when it comes to these kind of methods. Yeah, absolutely. I mean, we used it in that work. And when it comes to traceability, again, it's not, I'm not a machine learning expert, but I know that actually last week I was approached by a blockchain company that was really trying to say, look, we need to start thinking about blockchain in the realm of seafood traceability. Now, I'm going to be very honest with you. The seafood industry is quite reluctant to have outside companies come to them with either electronic tools or blockchain tools or whatever for improving traceability. Either they design their own because they're used to this kind of business-to-business system, or they just use paper. So there is also a change of mentality that needs to happen in order to incorporate some of these tools, and well, hopefully it will happen, but things change slowly. Thank you.
Yeah. One question there.
Thank you. Hello, Jonathan White. I work in Ireland at the Marine Institute. Marine, that was really interesting. I don't do much pipette science and I've forgotten a lot about it, so that's really nice to hear that. I thank you for that. A concept and a question. I believe that the majority of human beings want to do the right thing. They want to abide by rules and regulations. And I see what you're looking and thinking at here is a monitoring tool. For ecological science, it's wonderful. For the kind of work that you're looking at, I wonder if we would be more productive if we focused at understanding why the fishing community is doing what it's doing to not abide by regulations and trying to work with them rather than trying to point the finger at them. Thank you.
Yeah, thank you. What I want to just point out here is that, so I talked, for example, quite a bit about mislabelling and how some of these tools can be used in the context of enforcement. If we're talking about fraud here, which seems to be what you're talking about, it is not necessarily something that happens at the level of the fishing community. It does, and that's why I did give that example earlier in the talk. But in fact, we don't necessarily know at which level of the supply chain it happens. It could happen very late in the supply chain. It could happen at the level of the export, for example. So that, I think, would be the first question to answer. Where is this fraud happening? Is it really the fishing communities, or is it somewhere else, right? I can tell you that wholesalers, retailers, even fishmongers probably also partake in this. So we have to find where the problem is in the first place. And then, of course, when we know, which at the moment we don't really know, that's the truth of the story. But if we have more clarity on this, then of course trying to understand why this happens in the first place would be useful. But it's true that if this deceit happens for financial gain, I mean, I'm not a criminologist here, but you need to also ask yourself at some point, where do you just use enforcement tool to stop this, and where do you just communicate to try and make things better? Like this, but yes, as you said, they are somewhat philosophical considerations.
Thank you.
Thanks.
Thank you very much.
Thank you so much for a really engaging talk. I'm David Murray, CFAS, and I was really interested to see the slide on the whiting variants, the 3 different variants that you found in the Celtic Sea using the fast fish ID. I was wondering if you could expand on that a little bit, and if you had any additional information on whether it was all in the Celtic Sea or was there any regional differences you noticed beyond what you presented in your presentation.
Sorry, you're talking about the 3 different variants?
Yes, in the
whiting?
mitochondrial.
Yeah. Well, I can tell you an interesting story, actually. When we were on that boat, it just so happens that we got a lot of signatures for anchovy. Many different signatures. It was terrible because obviously it seemed like the method worked well for most species, but there are some species that have so many variants with it, that mini barcode, that the method just becomes completely useless. However, we did Sanger sequence these individuals, and what we realized, interestingly, is that that mini barcode is placed exactly at an area within the CO1 that can tell apart 2 different clades of anchovies. And we actually got pretty strong signatures between 2 different clades of anchovies, one that is pretty typically found in these more northern regions, and one that is more southern. And we could see that we had some individual from the more southern clade that were there, but in lower abundance. So, and that was quite exceptional because the method is absolutely not designed to be able to see different clades or where your fish comes from. But as a matter of fact, for anchovy, we were able to see that. But yeah, other than that, the variant itself can be a problem if you are dealing with a species that has a lot of variants. But generally speaking, the company chose these mini barcodes because they tend to be quite similar within a species and different between species. So yes, but it can be an impediment to the universality of the method for sure.
Great, thank you so much. I think there was a good few more, well.
Thank you, Maureen. My name is Dorothy Dunkel, and I work at Sintef Ocean in Bergen in Norway. And when listening to you, thank you so much for your talk, first of all. When listening to you, I get the feeling also that the burden of proof should be turned. It shouldn't be our job or enforcement's job to always find the bad guys. The producers should prove where, you know, at the grocery store where their stuff comes from. And it reminds me of a fish hackathon that we had at one of the John Kerry's international fish hackathons. And one of the groups in Bergen came up with an app where they could take pictures based on machine learning on the myomeres, the muscle tissue of these, and correlate that with species that have a high probability of being found in that area, which puts the power back to the consumer to say, demand for this and increasing ocean literacy. That was also part of your talk. We have such low ocean literacy. So I was wondering what your reflections are about turning the burden of proof back to the producers.
Yeah, I mean, that's a very good question. I don't know if I really have an answer to it. I think, unfortunately, it should be the producer's responsibility, but I don't know to which extent it should be their responsibility, in fact, to prove that the products, let's say, comes from a particular region. It's a system that, in a way, depends a lot on trust as well, right? So the idea is that your producer, your fishers, etc., are indeed going by the regulation, they are indicating where the product comes from, what the species is, and this is truthful and accurate all the way up to the consumer, and there's nothing else to it, right? The problem is that we do know that there are some errors in the traceability system that lead to these kind of mislabeling, and that, of course, there is also fraud, but that's not really something that the industry necessarily wants to acknowledge. So I think it's hard to tell them. I mean, we could try, but it's a difficult place to be in, a difficult question. But yeah, I'm not sure I understood the question super well, but maybe we can talk about it again afterwards.
Trust has to be earned. They have to earn the trust. And I think there's a niche for business to have wholesaler seafood producers that say, you should trust us. This is how we show it. And I think, you know, trying to look for those producers and those, you know, fishing organizations that want to build up trust with these methods, they should pay for the methods, not us. Because this is their product that they want to have a good, trustful relationship with their consumers. That's my opinion.
Mm-hmm. Yeah, and just to add on that, it's a very, I mean, it is an interesting point. And at the moment, the European Commission is considering creating a sustainability label system for food products in the EU, and possibly as well for seafood products. We don't know whether it will be mandatory or voluntary, probably voluntary. But the big question that everyone has here is, who is going to measure the sustainability of these products? Is it going to be the industry? Is that something the industry should do themselves? Is it going to be you, the Commission? Is it going to be the retailers? At which point of the supply chain is this going to happen? So these are indeed considerations, even for something like that sustainability label. And interestingly, I don't think anyone has the answer to it. The industry doesn't want to do it because they think it's too burdensome, and we're asking too much of them. And the Commission kind of goes along with that a little bit and says, don't worry, we'll be the ones doing it. So yeah, maybe it is a mentality we need to change as well. Thank you.
Okay, I think there are more questions.
Yes, this one.
Okay, one more.
Yes, very short. I am Pablo Gonza from Spanish Institute of Oceanography, and thank you very much for the nice and interesting presentation. It's just only if you have information on the application of these techniques and systems for the identification of fins, sharks fins, identification of the fins of sharks in the markets, international markets, and if you see that this can be applied in a very cheap and effective way in those markets or fisheries. Thank you. This is about sharks, about the commercial of fins of the sharks. Just, I asking if you have information on the application of these techniques to identify the sharks, the species of the sharks, just in these fin markets. Okay.
Yeah, I mean, what kind of information are you interested in?
No, just to know, because there's a big problem in the international markets just with these species. And then one of the systems will be just to identify clearly the species, and not just to take all the sharks in a bag, just to identify by species on this important market in China and in Japan and so on. So, yeah.
So yeah, I mean, what I can tell you when it comes to the shark export from Indonesia, as I was mentioning earlier, it's one of the biggest exporter of shark products, particularly fins. They export a lot to Hong Kong and to Singapore. There's an interest both from the exporting side, so Indonesia, and the importing side, so in this case, Hong Kong, Singapore, to test what the species are. The problems is there are quite a few CITES listed species that find their ways into these exports, and they should not be exported. There's a lot of shark products that are being exported, lots, and it is hard to test them all. They're kind of all mixed up in these big batches, and then they're just sent over. if there was some sort of regular system where the products were tested upon departure and upon arrival, it would maybe strengthen a bit the process and prevent these types of products from being traded. But it is very difficult. And occasionally they do catch entire batches of a CITES listed species and they just retain that and prevent it from being exported. But yeah, it's, I mean, you need a whole system to be in place. And again, it's a complicated endeavor. Yeah, thank you. Thank you very much for the questions.
Okay. Thanks so much again, Maureen. This was amazing. And yeah, I think there was a lot more questions to be had, so go for it, Maureen. And yeah, we also have, thank you for your, thanks again.